Hi everyone, my name is Helen and I am a master's student in the Kisakata 2 lab. Today I'm going to talk about my thesis project on developing coding for platelet storage devices. As you all probably know, platelets are cells in our blood that stops bleeding. And for patients with a low platelet count or poor platelet function, they require transfusions to prevent or treat their bleeding. However, platelets have a short shelf life, which leads to a lot of waste and a lot of shortages. Currently, platelets are stored in plasticized PVC containers at 22 degrees Celsius for a maximum of 5 to 7 days. Their short shelf life is attributed to two main causes. First, platelets undergo deleterious changes in structure and function over their storage period due to various mechanisms. This leads to decreased circulation time and hemostatic activity after transfusion. Storage at room temperature also makes platelets susceptible to bacterial growth, as the conditions are ideal for many bacterial strains. There are currently two main ways to prevent bacteria contamination. Preventative measures such as donor screening, removing the first aliquot, and regular testing are generally effective at preventing bacterial contamination. However, the process of blood collection, processing, and testing for pathogens is time-consuming, and platelet quality continues to deteriorate during this time. In addition, even with these measures in place, 1 in 2,500 contaminated units actually escape detection, leading to adverse transfusion reactions. Pathogen inactivation technologies represent a newer, more proactive approach that uses UV light to target the nucleic acid of pathogens. Although this addresses the gaps in the preventative measures, UV light is nonspecific and may cause damage to platelets, resulting in decreased circulation time and hemostatic activity after transfusion. A third approach to address this problem is to modify the platelet bag surface, since bacteria and platelet adhesion is strongly dependent on the physical and chemical properties of that surface. The current hydrophobic PVC surface is not only conducive to bacteria and platelet adhesion, but also the activation of platelets. By adding a hydrophilic non-fouling coating, we could address the problem of bacterial growth and platelet adhesion while also saving the time required for the testing. And this brings me to the objective of my project. To do this, we will use the interaction between polydopamine and known hydrophilic polymers to coat the platelet bags. And then we will incorporate antimicrobial peptides to actively kill any bacterial contaminants. Using this method, we performed some preliminary platelet adhesion tests on different polymers. We first coated the platelet bags using the hydrophilic polymers and polydopamine, cut the bags into coupons, and incubated them with platelets. After washing to remove any unbound platelets, we added detergent to lyse the cells and incubated it with an LDH catalyst and dye to quantify the number of platelets adhered to the surface. Here are the results of 5 different polymers at polymer dopamine ratios of 5:1 and 15:1. And based on this data, PDMA, polyacrylamide, and PMPC at ratios of 15:1 reduce platelet adhesion by over 90%. Building upon the antifouling background, we added AMPs to the PDMA coating and incubated them with bacteria. Preliminary tests show that a PDMA coating with the antimicrobial peptides E6 and TET20 led to a 100% and 90% reduction in bacterial growth compared to the control, respectively. Next, we plan to expand the screening to a library of antimicrobial peptides on different antifouling backgrounds and assess its antibacterial activity in complex media. If you want to learn more about the effect of the coating on platelets, you can check out Nicholas's poster at poster number 2. And I would like to thank my our collaborators, my committee members, and everyone in the Kisakadatu lab for their support. Thank you.
I think we could have we have time for some questions if anybody has a question.
Hi. Hi. Why dopamine?
Dopamine, we chose to use dopamine because dopamine is inspired by muscle adhesive protein. So dopamine is biocompatible, and it can adhere to a wide variety of surfaces. So it will be able to adapt to the different materials that we use for blood bags.
Nice talk. I'm wondering, obviously it's important that the platelets aren't activated in addition to not sticking. So what sort of tests are you planning to run for platelet activation?
You can check out Nicholas's poster.
All right, will do.